Today we're going to talk about protein structure and function, and the goal of this video is to explain how the sequence of amino acids in a protein determines a protein's shape and function. So, the amino acid sequence of a polypeptide is only the first step in making a functional protein. The amino acid chain has to fold up into a specific shape, and it's that specific shape that gives the protein a specific function. So it's important to understand the parts of an amino acid. So on the right here, I've put a picture of an amino acid, and you'll notice there's an R at the bottom. This is called the R group. It's a side chain, and it's that R group that makes each amino acid unique. So when you look at a codon chart and you see the names of the different amino acids, what makes each of those different is what's attached right here where this R is. Amino acids have these other parts in common. So on the left in blue, you have an amine group, and on the right in pink, you have a carboxyl group. So every amino acid is going to have an amine group and a carboxyl group. Those will be identical. But the parts that make each amino acid unique is the R group. Now that's going to become really important because it's going to play into how a protein gets its specific structure and function. Okay, so here I've got a polypeptide, which is just a string of amino acids together. And it's the interaction between the amine group and the carboxyl group and the R groups that determines the protein's structure. So when you look at this polypeptide, here is the first amino acid. Notice I have an amine group, the C with the double bond O, that's my carboxyl group. And in this case, the R group is just an H. So that's the first amino acid in this polypeptide. Here's the second one. Here's my amine group. It's got the nitrogen, my carboxyl group with the C double bond O. But this time the R group is different. Instead of an H, it's CH3. So this is a different amino acid. This is why it's amino acid number 2. You keep going, amine group, carboxyl group, a different R group. So this is amino acid 3. Amine group, carboxyl group, and here's a fourth R group. So I have 4 amino acids right now in this polypeptide. And like I said a moment ago, it's going to be the interaction between the amine groups, the carboxyl groups, and the R groups that gives the protein its specific structure. All right, now there are levels of structure in a protein. There's actually going to end up being 4 total. So the first level we call the primary structure. And this is just the sequence of amino acids in the polypeptide chain. So you just list in order what the sequence is of amino acids. That's the primary structure. The secondary structure is the way a polypeptide chain will fold in a repeating arrangement to form one of 2 things, one of 2 shapes. Either what we call an alpha helices or beta pleated sheets. If it's an alpha helice, then it will be shaped like a spiral. So you'll notice here, this N, that's the amine group, and this C double bond O, that's the carboxyl group. So when that amine group and that carboxyl group form a bond, it gives it this spiral shape. Here, you've got the amine group and the carboxyl group, and they can form a bond. But notice in this case, the bonds are they're parallel to one another. They're going to be directly across from one another. So these 2 groups can interact in 2 different ways. They can create spirals, or they can create these flattened folds. The spirals are the alpha helix shape, and then the flattened folds is the beta pleated sheet shape. Okay, so these 2 shapes, those are the 2 types of secondary structures that the protein can have. So you started with a chain of amino acids, and based on how the amine groups and the carboxyl groups in that chain interact, it can either form an alpha helix or a beta pleated sheet. So these are the secondary structures. The tertiary structure is the way that those secondary structures interact to form a more complex shape. And specifically here, it's going to what ends up giving the protein its tertiary structure is largely going to be the interaction of the R groups. So that's what you're seeing here on the left. If the R group has two S's, that can form a disulfide bond, or a hydrogen bond could be formed, or the interactions could be hydrophobic, which means it doesn't want to mix with water, or it could be hydrophilic. It does want to mix with water. So there's lots of possibilities here about how the R groups will interact with each other, but it's those interactions that's going to give the protein its tertiary structure. And so this is when you start getting a more complex shape. So in this case, I see spirals, so I know that the secondary structure was the alpha helix. But now looking at this, we start to see how each spiral can fold in different ways based on how the R groups interact to give it this more complex shape. All right, we get to the fourth and final level here. So the quaternary structure of a protein is the way that multiple polypeptides interact to form a single functioning protein. So the tertiary structure is still dealing with only one polypeptide. But when you start putting multiple polypeptides together, and that's what you see here on the right, I have at least one pink polypeptide. It looks like there might be two actually. And then I have at least one, possibly two, yellow polypeptides here. Notice we're putting more than one polypeptide together. And when you start doing that, you are dealing with the quaternary structure of a protein. So these are the 4 levels of structure in a protein. And this gives you a kind of one picture summary. So you start with just a sequence of amino acids. That was the primary structure. Then those amino acids, specifically the amine groups and carboxyl groups, interact in certain ways. And that's what gives it the secondary structure. The 2 possible secondary structures were alpha helix or beta pleated sheets. And then those secondary structures that you have will interact in different ways based on the R groups. And that's what gives it its tertiary structure. And then when you put multiple polypeptides together that have these tertiary structures, that's when you get a quaternary structure of the protein. And the big idea is at that point, you have a very specific shape of the protein, and that very specific shape allows it to perform a very specific function in the cell.